Hello, everybody. My name's Tim Crook. I'm an oncology consultant with a big interest in melanoma and choroidal melanoma in particular. Thank you for asking me to contribute to the meeting. I'm going to give a short presentation on how precision medicine may contribute to diagnosis and management of choroidal melanoma. What do we mean by precision medicine or precision oncology? Well, it can be summarized in this short sentence: the right drug given at the right dose to the right patient at the right time. So it's essentially a way of optimizing and personalizing treatment based on the individual features, properties, or characteristics of an individual cancer, in this case, a choroidal melanoma. We already do it in cutaneous melanomas and in mucosal melanomas, of course. We do BRAF testing routinely with highly potent small molecule inhibitors of mutated BRAF, which is present in a substantial minority of cutaneous melanomas. We also do it in mucosal melanomas, where mutations in the c-KIT gene can be targeted by imatinib or nilotinib. We don't have actionable driver mutations at present in choroidal melanoma. Although clearly that's an area of active research interest. It's worth asking at this early stage, why do we need precision medicine for choroidal melanoma? Well, firstly, as we'll all be very well aware, there's very few, in fact, zero proven effective systemic therapies for metastatic disease. Immunotherapy can be helpful in some patients, but has limited efficacy. There's no licensed targeted systemic therapies. No confirmed effective adjuvant systemic therapies after treatment of a primary lesion. And as we'll all be aware, previous trials of cytotoxic chemotherapy in unselected or non-profiled patients have very disappointing response rates. So clearly we need to improve. We need to be able to offer our patients better systemic therapies. How do we create a precision medicine profile? Well, we can do it from tissue, where we can either profile the primary tumor or a metastatic lesion, typically in the liver in this disease. But for choroidal melanoma, sometimes we don't have tissue from the primary. If the tumor is treated with ruthenium plaque radiotherapy or proton beam therapy, it's often not biopsied. And so we don't have tissue available from the primary to make a precision medicine profile. And further, sometimes we can't get adequate tissue from a metastatic lesion in the liver. Sometimes we can, sometimes we can't. What do we do if we can't get tissue? Is that the end of our hopes to generate a personalized profile? No, we use a liquid biopsy. We use blood. We use the peripheral blood supply of the patient. So we can define a liquid biopsy as a test done on a sample of blood or body fluid to look for cancer cells from the patient that are circulating in the blood, or for pieces or fragments of DNA from tumor cells that are circulating in the blood. Now that's a kind of generic definition and helps to guide us what we're trying to achieve with a liquid biopsy. A broader definition will include some of these parameters: circulating tumor cells, cell-free DNA, exosomes, which allow us to measure gene expression, and microRNAs. It's a very appealing thing to do for a clinician. The liquid biopsy can be valuable in early diagnosis, prognosis, treatment selection, monitoring of treatment effect, and detection of early recurrence. In other words, diagnostic, predictive, and prognostic biomarkers can be readily obtained from a small sample of peripheral blood. So it's a potentially exciting technology, and one that's very underused, in my opinion, at the moment. Invasive biopsies, in other words, tissue biopsies, are challenging sometimes. They're expensive, they're invasive, and they're prone to complication. The patient with ocular melanoma will frequently have liver metastases as the first manifestation of metastatic disease, and a liver biopsy is not a trivial undertaking. Perhaps more importantly, from the mechanistic perspective, tumor heterogeneity worries us that sampling a single clone may not give representative analysis of what is a heterogeneous tumor. So popping the liver biopsy needle into that one part of the liver and using that to inform treatment decisions may not be representative of the wider tumor. There may be insufficient number of cancer cells to carry out meaningful, adequate downstream analyses. So in other words, the biopsy is inadequate. Formalin fixation, if we're using archival tissue, can and does introduce significant artifacts and can have limiting effects on the use of RNA, for example. So there are challenges with invasive biopsies, and as we noted in the previous slide, a liquid biopsy affords one option to overcome that. Here's the sort of things that we can do from our tube of blood. We can find multiple DNA abnormalities. We can look at RNA expression and fusion transcripts in exosomes. We can look for tumor emboli. We can detect circulating tumor cells. We can find and analyze circulating tumor DNA and look for mutations. And we can find, and those DNA changes can be at the point mutation level, translocation and chromosomal abnormalities, deletion or amplification of areas of chromosomes. So we can get a vast amount of potentially very useful information from a blood sample now. Part of the analysis of that will be using what is known in the trade as NGS, or next generation sequencing. Used to be called high throughput sequencing, sometimes called massively parallel sequencing. It essentially allows the laboratory to sequence multiple times, hundreds, thousands, tens of thousands, hundreds of thousands of times, each individual fragment of DNA. And this is important in a liquid biopsy in particular, because the liquid biopsy will the cancer cell DNA will frequently be diluted by normal peripheral blood DNA coming from normal tissues. And it's only through next generation sequencing that you can pick up the needle in the haystack, as it were, the mutated fragment of DNA amongst the large background of non-mutated. So it's been a massively important technological advance, allowing performance of detailed DNA analysis, mutation finding from peripheral blood. High throughput, systematic, unbiased mutation detection. And with digital PCR, you can get a very straightforward readout in a well-equipped lab. The way to do liquid biopsies, precision medicine, in my view, and I'll show you some of the evidence for it in a moment, is to combine this very powerful technology of next generation sequencing with other parameters that allow us to generate a comprehensive profile of an individual patient's cancer and thereby make informed treatment decisions. And I want to describe the platform that I use, which is called Exacta. It's blood-based, in other words, from liquid biopsy. It incorporates multiple analyses and feeds out like a sausage machine into a treatment recommendation. The platform combines multiple parameters, including next generation sequencing, to identify appropriate, potentially useful treatment regimens for individual patients. It looks at DNA, it looks at RNA, it looks at tumor cells, it looks at immunocytochemistry to discover all possible targetable alterations present in the cancer. It evaluates biomarkers associated with prediction of immunotherapy benefit, targeted therapy benefit, and also affords a readout of the hierarchical efficacy of cytotoxic drugs. What that means in plain speaking is that it will tell you which are the sensitive drugs and which are the useless drugs. So it gives you an integrated plan of how a cancer might respond to the treatments available to us. And these include standard of care and off-label therapies. Now, clearly for a choroidal melanoma, there are very few standard of care options, and most of the therapy recommendations are likely to be off-label, but this should not discourage us. This is a summary. This slide's slightly older now because the number of tumor DNA single nucleotide variants, SNVs, is more than 453 genes. There are more than 51 genes being searched for rearrangements and amplification. 22,000 protein coding genes are analyzed in exosomes and by RNA sequencing. and up to 30 drugs and or combinations thereof can be evaluated in real time in vitro chemotherapy sensitivity on live tumor cells. The turnaround time is 7 to 10 days, and the technology platforms are detailed there. So it's really a very comprehensive, integrated way to look at an individual cancer in real time and try to derive therapeutically useful information from it. Exosomes, as I've mentioned, you can look at cancer cell gene expression in peripheral blood without having to sample the tissue directly, using exosomes, small vesicles, tiny vesicles released by a cell, which contain the RNA and proteins, and you can pick them up easily in blood and analyze them. A very useful intervention. I want to talk about things now called CTACs, which form part of the liquid biopsy using this platform. CTACs stands for circulating ensemble of tumor-associated cells. They're a very novel finding, only published this year. They're a hallmark of cancer. A CTAC is actually not a circulating tumor cell, CTC. It's a circulating tumor cell with its associated stroma and support cells, e.g. macrophages, e.g. fibroblasts. You can pick these up with appropriate technology in very large numbers in peripheral blood, much higher than circulating tumor cells. They're ubiquitous and present in all solid tumor types thus far looked at. CTACs can be used for diagnosis and particularly for profiling drug sensitivity. They're extremely relevant for melanoma, including choroidal melanoma, where, as we discussed at the beginning, sometimes a tissue biopsy is not possible. So CTACs, circulating ensemble of tumor-associated cells. This is how you isolate them. You take a tube of peripheral blood, you add it into the lab in the presence of what's called an epigenetically activated tissue culture medium, which induces paradoxical apoptosis. What that means is that all normal cells in the peripheral blood are killed by apoptosis. They die. The normal tissue, therefore don't contaminate the background. And you can grow out by day 5 these clusters. And you can see in this little pictures here that the gray view is taken 5 days after establishment of the CTAC culture. You can see that almost all the background cells have died away, and there are 2 large clusters there. These are CTACs, so these contain viable, living, proliferating cancer cells from peripheral blood. Because they have their support cells with them, they will proliferate at much higher numbers than circulating tumor cells. And the isolation is very gentle because it uses cell biology and paradoxical apoptosis to facilitate generation of pure cultures of cancer cells. And you can characterize them here. We see the use of DAPI, EpCAM, CK, CD45. For melanoma, we'd use different markers, but these are just an example. And by day 6 in culture, you start to get organotypic or organ-specific markers showing up on these cells. So they're extraordinarily interesting and extremely useful. And here's some clusters from various patients from peripheral blood. There's 12 shown here. And the white arrow in each case points to the enlarging cluster. And in all cases, you can see that the background normal cells are in the process of dying or have died, allowing the outgrowth of CTACs. And within a week, you can get ample CTACs for immunocytochemical analysis, for functional analysis, and for in vitro chemosensitivity cytotoxicity testing. And one such assay here, this is chemotherapy sensitivity. Simplified view, on the top we have, these are CTACs growing over 12 hours. The green, yellow, fluorescently stained cells are CTACs, and they've been treated in the first row down with 5-fluorouracil. These are breast cancer CTACs. The principle is identical. And below Taxol. And you can see that these CTACs are very sensitive to 5-fluorouracil or capecitabine. And in the bottom line, the CTACs are sensitive to the drug Taxol or paclitaxel. An absolutely identical experimental plan is used for CTAC analysis in choroidal melanoma. And after a week, you get this kind of detailed report, which will tell you, combining all the parameters looked at in this platform, will tell you drug sensitivity, gene expression profiling, DNA alterations, immunohistochemistry, drug resistance biomarkers. You'll get a recommendation, treatments which might work in green, treatments which certainly won't work in red. And this then is fed back to the requesting oncologist, who then has to use his oncological knowledge and sit down with the patient to discuss the options that have been identified. And you'll get this hierarchical stratification of sensitivity. In this case, a breast cancer patient, we see that the drug eribulin is the #1 pick. And this is because 70% of the CTACs were killed within 24 hours. And this is the cutoff. This is the time point at which cytotoxicity and drug sensitivity is measured, reproducibly shown to be the best correlate of in vivo, i.e. clinical responsiveness. And the clinician will then sit down, study this, and sit down later with their patient and come up with an appropriate treatment plan for that patient. This is the first proof of principle clinical study, this waterfall plot. Along the left, we see the vertical axis is changes in dimension of the tumor according to RECIST criteria. Anything above the line, above 0, has got bigger. Anything below the line has got smaller. The individual colors represent the cancer types demonstrated here. So this is very much a cancer agnostic platform. And you can see that in the pale yellow, there's several melanoma patients in there with encouraging results. So 126 patients were evaluated in this proof of principle, and you can see the utility, the clinical utility of these multiply pre-treated patients in terms of their clinical response. Now, I just want to show you how this has worked in practice. This is a 53-year-old patient, previously fit and well, who had a choroidal melanoma in his left eye 6 years ago. This was treated with orthodox proton beam radiotherapy, and the patient then went on to standard surveillance with 3 monthly liver function and clinical checks and 6 monthly non-ionizing liver imaging. He remained well for 3.5 years until routine surveillance picked up liver metastases in October 2017. A rebiopsy was possible in this case, and the metastatic disease was confirmed to be choroidal melanoma. He was treated with combination ipilimumab-nivolumab, followed by nivolumab monotherapy, and did remarkably well, staying almost completely stable disease and healthy for 2 years. At that time, his blood markers, serum S100 and LDH, were rising. His liver function was beginning to deteriorate, and liver MRI confirmed the presence of progressive disease in the liver. He underwent ablation of the liver metastases in September, but had further progressive disease in January this year. At that point, he undertook liquid biopsy with multi-analyte analyses, as I've just described. His mutational load was 17.03%. So that is the proportion of the DNA in the peripheral blood that is derived from the cancer, in this case, the choroidal melanoma in the liver. With 2 classic mutations, GNA11 and SF3B1, both well-recognized choroidal melanoma mutations, validating this as a very sensitive technology for picking up disease. The in vitro cancer cell sensitivity, so there were no targeted mutations, unfortunately, unsurprisingly, from the DNA analysis, but in vitro cancer cell sensitivity analysis showed a number of drugs to which this patient's CTACs, i.e. the cancer cells we were sampling in the blood, which were representative of the cells in his liver, were sensitive. And you can see that both oxaliplatin and carboplatin had similar sensitivities, which is encouraging. Two drugs from the same mechanistic group were effective. And he went on to receive 5 cycles of carboplatin, stabilized his disease, improved the liver function, and he remains clinically stable. And we hope that this treatment will bridge him through to the availability of IMC-GP100 as he's HLA-A2 positive. So there's an example there where this has been really very valuable and given this gentleman a new lease of life, and hopefully, potentially, will allow him to stay well through into next year when we may have a new therapeutic approach for him. There's another application I just want to briefly touch on with CTACs. Can we use molecular biology for surveillance? I referred there to the orthodox choroidal melanoma surveillance program after treatment of the primary in the eye, which is essentially liver function tests and liver imaging. We tested CTACs in 16,134 members of the public, 10,625 of whom were asymptomatic and healthy. 5,500 had epithelial cancer, and we measured their CTAC levels. Remarkably, the CTACs were present in over 90% of the patients who had cancer, proven cancer, but were rare, 3% amongst the asymptomatic population. The members of the asymptomatic population who had positive tests for CTACs have been followed up closely for a further 12 months and have a 230-fold increased cancer risk within that 1 year compared to individuals who did not have CTACs at baseline. Now, for us, this is an extremely important and significant finding. It implies that the presence of CTACs in peripheral blood mandates a close oncological or clinical follow-up. Critically, CTACs can be further analyzed to identify the primary site with selective immunocytochemistry. And so we can tell those 3.6% of high-risk patients where the likely primary is and what sort of cancer it is. And you can imagine that that is extraordinarily helpful for any cancer type, but we're now going to test this in melanoma to see if CTACs and circulating tumor DNA, circulating free DNA, is a more sensitive, a better, a more cost-effective, a more patient-friendly way of both finding primary disease and surveying, keeping our patients who've had their ocular melanomas or their cutaneous melanomas or their mucosal melanomas treated in the primary setting, excuse me, whether this represents a better way to keep them under surveillance, a more sensitive way to identify early relapse and intervene earlier. So in conclusion, in my opinion, personalized precision oncology, whilst it's certainly in its infancy for choroidal and other types of melanoma, it is going to be a very important and emerging technology for improving clinical outcomes, which is the only endpoint all of us want. Despite the early days, we can see that comprehensive profiling can shed new light on the properties of an individual patient's disease and identify potentially clinically useful, effective therapeutic options. ctDNA, circulating tumor DNA, aka circulating free DNA, is a very promising modality for population surveillance and indeed for keeping the treated patient population closely monitored. combined with CTACs, circulating ensembles of tumor-associated cells, for me, this offers unique opportunities for tumor-specific monitoring, both of the patient population, the treated population, and indeed for the general population, for detection of early subclinical disease and surveillance of the treated population for detection of early recurrence or metastasis. That's my talk. Thank you very much for asking me to contribute to the meeting.